Our final SoftChalk lesson in Unit 1, Lecture 5 deals with bacterial pili and fimbria. Let's look at our fundamental statements for this lesson. Fimbria and pili are thin protein tubes originating from the cytoplasmic membrane, and they're found in virtually all gram-negative bacteria, but not in many gram-positive bacteria. Pili are typically longer and fewer in number than fimbria. The short attachment pili, sometimes called fimbria, are organelles of adhesion. They allow bacteria to colonize environmental surfaces or cells and resist flushing. Antibodies made against bacterial pili may block colonization and or promote opsonization. The long conjugation pilus enables conjugation in gram-negative bacteria. The pilus has a shaft composed of a protein called pilin with an adhesive tip structure at the end that has a shape that corresponds to specific receptors on host cells. The same bacteria may switch the adhesive tips of pili in order to adhere to different types of cells and to evade immune defenses. And finally, type 4 pili not only allow for attachment, but also enable a twitching motility that enables the bacteria to crawl or walk over the surfaces to which they've attached by extending and retracting their type 4 pili. And now let's look at our detailed learning objectives for this SoftChalk lesson you need for the exam. You should be able to state the chemical composition, structure, and function of the short adhesion pili in bacteria. Briefly describe how antibodies made against the adhesive tip of bacterial pili may block colonization and or promote phagocytosis. State the function of a bacterial conjugation or sex pilus. Define bacterial conjugation. State how the ability to change the shape of the adhesive tip of its pili could be an advantage to a bacterium. And briefly describe twitching motility induced by type 4 pili. And we have one highlighted bacterium in this SoftChalk lesson, Neisseria gonorrhoeae, which causes gonorrhea. So let's start out with the structure and composition. Pili and fimbria are thin protein tubes. They originate from the cytoplasmic membrane of many bacteria, and both are able to stick bacteria to surfaces, but pili are typically longer and fewer in number than fimbria, but we often do use them interchangeably, the terms. It's also important to realize that pili are found in virtually all gram-negative bacteria, but very rarely in gram-positive bacteria. Fimbria and pili have a shaft composed of a protein called pilin, but at the end of the shaft is an adhesive tip, a sequence of amino acids that has a shape that will bind to specific glycoprotein or glycolipid receptors on a host cell. And we see this in the illustration in Figure 1. So this is the shaft of the pilus composed of pilin, but then eventually a sequence of amino acids is transported down and stuck to the end called the adhesive tip. And the order of amino acids determines the shape of that tip, and that shape allows it to attach to receptors on host cells so that they can resist flushing. There are two basic types of pili. The short attachment pili, which are also known as fimbria, are usually short and very numerous, and they enable bacteria to colonize environmental surfaces such as host cells and resist flushing. Whereas long conjugation pili, sometimes called F+ pili or sex pili, are longer and fewer in number, and they of course enable conjugation. And remember, conjugation is genetic recombination where there's a transfer of DNA from a living donor bacterium to a living recipient bacteria by cell-to-cell contact. And in gram-negative bacteria, that would involve a sex pilus or conjugation pilus. And Figure 3 shows an illustration of a bacterium having numerous short attachment pili and producing a conjugation pilus or sex pilus that's able to contact another bacterium for genetic recombination. And this is a movie that shows some conjugation pili. The bacteria are labeled in red, and the conjugation pilus is in green here. So there we see some long conjugation pili in green. And if you watch them, you do see that some of them are retracting or depolymerizing and then reforming. And here we see one that's making contact with another bacterium. There's one that's retracting that didn't make contact. So these pili are constantly forming and retracting. By retracting, we mean they depolymerize or pull themselves in. And this is a nice movie that shows you a conjugation pilus or sex pilus retracting. So there you see in green the conjugation pilus, and you see it getting shorter and shorter as it depolymerizes or retracts. And if that were to contact another bacterium, that would pull the two bacteria together so that a mating pair can form. And there's a little self-check on the structure of bacterial pili and fimbria. Now we're concerned about the significance of pili to bacterial pathogenicity, the ability to cause disease. And as we said, the short attachment pili, also called fimbria, are organelles of adhesion. So they allow bacteria to colonize environmental surfaces such as host cells in the body and thus resist flushing. Remember, virtually all parts of the body have a flushing action. And so if a bacterium is going to colonize the body, the first step to causing disease or becoming microbiota, they have to attach to cells. Now, we've already seen in previous SoftChalk lessons that most bacteria use cell wall adhesins, surface proteins on the cell wall, to adhere. Bacteria often use a glycocalyx to adhere, and gram-negative bacteria often use pili as well to adhere, to colonize and resist flushing. And again, the shaft of the pilus is composed of a protein called pilin. As we said, the adhesive tip has a shape that will fit a corresponding glycoprotein or glycolipid receptor on a host cell, as we see again in figure 1. Now, one of the advantages of pili might be because both bacteria and host cells have a slight negative charge. Remember, nucleic acids and many proteins are negatively charged. So pili may enable bacteria to bind to host cells without initially having to get very close enough where they could be pushed away by electrostatic repulsion. If the bacterium was very close to the cell, the negative charge of the bacteria, the negative charge of the host cell might push them apart. But the pili are much longer, and it's kind of like casting out a fishing line that has a hook on the end, and that allows it to attach to a receptor on the host cell, as we'll see in this animation down here. So the bacterium can initially make contact, and then the pili typically then depolymerize, and as they depolymerize, that pulls the bacterium towards the host cell, where cell wall adhesions and the cell wall adhesins can bind to adhesin receptors for a more intimate or tight contact. So if we look at this illustration here, Neisseria gonorrhoeae in this illustration is a good example of that. We see that it has pili, attachment pili with an adhesive tip on the end. In blue, we have the receptors for the adhesive tip of the pilus. And then it also has cell wall adhesins shown in brown, and the green are the receptors for the cell wall adhesins. So through motility or Brownian motion or movement of fluids, the bacteria may get close enough to a host cell where these attachment pili with their adhesive tip can bind to a receptor and resist electrostatic repulsion. So again, you can picture them as kind of like a long rod at the end with a hook on the end that can attach to a receptor. And then what typically happens is that as the pili depolymerize or shorten, they pull the bacterium down towards the cell surface. The bacterium kind of reels itself in so that cell wall adhesins can then bind to adhesin receptors. So here we see the pili depolymerizing, and that allows the cell wall adhesins to bind to adhesin receptors and adhere tightly to the host cell. So most gram-negatives use both pili and then cell wall adhesins to attach to cells. And bacteria are constantly losing and reforming pili, polymerizing them and depolymerizing them. And the same bacterium can actually switch the adhesive tips of the pili. There are often multiple alleles in the bacterial genome for both adhesive tips of pili and for cell wall adhesins. And that allows then the bacterium, by being able to make different adhesive tips, to adhere to different types of cells and evade immune defenses, which we'll talk about a little bit more when we get to bacterial pathogenesis. So for example, in figure 4, the bacterium on the left has this round adhesive tip that would fit this receptor on the host cell. So that bacterium in the population would adhere to that receptor on that host cell. But bacteria can change their adhesive tip by activating different alleles as pili reform, and they may put a different adhesive tip on the end of the pilin, which allows it to bind to a different receptor on a different host cell. And of course, as we said, most gram-negatives produce pili. Examples that we'll be talking about during the course, including Neisseria gonorrhoeae that causes gonorrhea, Neisseria meningitidis that causes meningococcal meningitis, uropathogenic E. coli that cause urinary tract infections, and Pseudomonas aeruginosa that causes a lot of opportunistic infections. And our highlighted bacterium here is Neisseria gonorrhoeae. So again, the importance of being able to adhere to cells, if we take something like the gonococcus, which adheres to the urethra, the tube that empties the bladder, if the bacterium lacks a way to attach, if it lacked initially pili, as the person urinated, the urine would flush the bacteria off the urethra. But if the bacteria have initially pili and then eventually cell wall adhesins to attach, that allows them to make fairly good contact with the cell so they can resist flushing by the urine in this case and continue to colonize and replicate in the urethra, causing inflammation of the urethra or urethritis. Now there's one class of pili we'll be talking about a fair amount during the semester, known as type 4 pili. These not only allow for attachment to host cells, but they allow a twitching motility. These are typically located at the poles of the bacteria, in the bacilli especially, and they allow for a gliding motility along a solid surfaces, such as over the surface of a host cell. So what these bacteria do is that at one pole, they extend their pili, which then attach with the adhesive tips, and then as they retract the pili, that pulls the bacterium or drags it along the surface till it reaches the adhesive tip. And then by extending and retracting and extending and retracting, they can continue to pull themselves over a surface. If they make pili on the opposite pole of the bacterium, they can change direction and move in the other direction. So we do see this gliding motility or twitching motility over solid surfaces very frequently in gram-negative bacteria. And it'll play an important role in pathogenesis in Unit 3. Bacteria can also use our type 4 pili for a slingshot type movement for a short distance over a cellular surface. In this case, the pili attach, and they attach from two different directions, and as the pili contract, they become very taut like a stretched rubber band. And as they get tighter and tighter, an anchoring pilus detaches, and the taut pilus then pulls the bacteria forward in a slingshot type motion in the opposite direction. And often we see changes between twitching motility and then the slingshot motility, which allows them to both do a rapid change in direction and be able to move over cellular surfaces. So let's take a look at how the type 4 pili can do this twitching motility or drag themselves over a cellular surface. We can picture them as crawling along the cell surface. So here we see a bacterium in this animation, and as it extends the pili, the adhesive tips attach, And then as it depolymerizes the pilin, that pulls the bacterium towards where it's attached. And then it can reform pili. The adhesive tips can attach. And again, as it depolymerizes, it keeps pulling the bacteria towards the attachment site. And this way, it's able to crawl over solid surfaces, such as over the surface of the host cell. And that way, it can spread out over the host cell as it replicates, and it can find better places for attachment. If it were to make pili at the other end, it can then crawl in the opposite direction. In the slingshotting motility, shown in this animation, we have the anchoring pili going out and attaching, and then they begin to retract, but because they're attached in different locations, these become very taut like a stretched rubber band. So as they retract, you get the pili stretching. And then as one pilus, the anchoring pilus, detaches, one of the anchoring pili, that causes the bacterium to slingshot over in a new direction. And we'll show you some movies of both of those. So this is Pseudomonas, showing its typical twitching motility over a solid surfaces. This is shown about 20 times its normal speed. And the box here is shown in higher magnification on the right-hand side. So, of course, Pseudomonas is a bacillus-shaped bacterium. You don't see the pili here because they're too thin to see with a conventional microscope, but as the pili extend and retract and extend and retract, that pulls the bacteria along a surface in a certain direction. If they make pili on the other end, then they can crawl in the opposite direction. So let's watch this. So here we see the bacteria crawling over a surface by extending and retracting the pili. Here's one going in the opposite direction by producing the other and then changes direction. So again, this is very important in allowing bacteria to spread out over a cellular surface once they attach. The type 4 pili attach rather loosely so that they can pull the bacterium forward and then extend and retract and extend and retract and crawl over surfaces. That's the crawling. What happens sometimes is when they produce the pili at a pole, as the bacterium divides and the pili are produced, the bacterium actually hops up on one pole, and then by extending and retracting the pili can actually walk over the surface. And there's a nice little movie of that showing the Pseudomonas walking over a surface. We're going to look at the bottom pair that have just undergone binary fission. And the one on the right is going to kind of pop up at the end once it produces the pili and begin to walk along the surface rather than crawl on its pole. So here's the bacteria getting ready to divide. And then it pops up on the end, and as the pili extend and retract, it actually walks along the surface rather than crawling. So you see both the walking and crawling motility very frequently with the type 4 pili. This is a very short twitching motility movie of showing type 4 pili from Howard Berg's lab. It's very short, but you'll see it extend a pilus and then the pilus pull it in this direction. And I'll play this a couple times because it's only a few seconds long. So there it extends and retracts. Extends and retracts. Get it one more time. Extend and retract and pull it in this direction. And here's another one where the bacterium is attached to the slide, but the pilus isn't attached to anything, where you can see the pilus retracting or depolymerizing. So there you see some of these pili retracting. Show it one more time. So as it depolymerizes, they retract. Now, if that was attached, it would be pulling the bacterium towards the attachment site. And this is a brief movie of the slingshotting motility with the type 4 pili, sometimes called hopping. where it's going to be pulled from one location to another as the pili become taut and then one anchoring pilus is released. So right now the pili are tightly retracting, and when one of them retracts on the top side, that pulls the bacterium down from where the arrow is to its new location. Now that allows for a quick movement, and a more viscous environment that would actually allow it to overcome the viscosity by being able to use that tension or rubber band action to pull the bacterium forward. Now let's look at the significance of Fimbria and pili in initiating body defense. So for initiating adaptive immunity, proteins associated with the bacterial pili, of course, function as antigens to initiate adaptive immunity. And as we've learned a number of times now, an antigen is defined as a molecular shape that reacts with both antibody molecules and with receptors on B and T lymphocytes. And we recognize an antigen as foreign when it fits the receptors on these B and T lymphocytes, and that initiates adaptive immunity. Now, pili are made of proteins, so this would be a protein antigen like figure 7a, and again, there could be many epitopes along that protein, and antibodies could be made to fit each of the epitopes along that protein antigen. So remember, the actual portion of that long protein antigen that actually fits the antibodies and fits the receptors on lymphocytes are called epitopes. In this case, anywhere from 5 to 15 amino acids with a unique shape that make up a portion of that protein antigen. And we have two branches of adaptive immunity. Humoral immunity is, again, the production of antibody molecules in response to an antigen, and that's carried out by B lymphocytes. And that's what we see in the case of pili. When antibodies are made against the adhesive tips of bacterial pili, they can prevent the bacteria from colonizing host cells by preventing the adhesive tip from binding to its receptor on the host cell. Also, if opsonizing antibodies are made against pili antigens, they can then stick the bacterium to phagocytes via the pili through opsonization. So figure 8a and 8b show you how antibodies made against the adhesive tip may block the bacteria from attaching to host cells and thus allow them to be flushed away. So remember, the adhesive tip at the end of the pilin, the shaft of the pilus, fits receptors on the host cell. That's how it locks in place. But if antibodies, as we see in figure 8b, are made to fit epitopes on that adhesive tip, they can cover up the adhesive tip so that it can't bind to pilus receptors on the host cell. And if those were opsonizing antibodies, they would then, of course, stick the bacteria to phagocytes for enhanced attachment or opsonization. And of course, our other immune defense is cell-mediated immunity involving cytotoxic T lymphocytes, activated macrophages, activated NK cells, and such. These are what we use primarily, though, to remove infected cells and cancer cells that display foreign epitopes and don't play much of a role in terms of bacterial pili. And there's a little self-quiz you can do on the whole SoftChalk lesson on bacterial Fimbria and pili.